Hello friends, how are you? I hope you might be safe at home. Home, stay safe. I am again back with sixth part of molecular basis of inheritance of 12th standard. Today's topic for discussion are transcription, transcription unit and gene, types of RNA and process of transcription. So let us start, students. First is about transcription. We have already studied in central dogma in the previous session that transcription is the process of copying genetic information from one strand of DNA to RNA. Now, the question arises, why only one strand of DNA is copied into RNA? The answer to this question is that if both the strands of DNA acts as template, they would code for RNA molecule with different sequences, and in turn, if they code for proteins, the sequence of amino acid in the protein would be different. Hence, one segment of DNA should be coded for two different proteins, and this would complicate the genetic information transfer machinery. Also, if the two strands RNA molecule produce complementary to each other, they form double-stranded RNA. So, the process of translation will not take place. Why? Because you have seen that if it will be double-stranded, so the translation process will not take place. Now, the next topic is about transcription unit. Transcription unit consists of three regions, that is a promoter, the structural gene and a terminator. As you all know that the two strands of DNA have antipolarity, means if one strand is from 3 to 5 end, then other will be from 5 to 3 end. Since the DNA-dependent RNA polymerase enzyme also catalyze the polymerization in one direction, that is 5 to 3 end. The strand that has polarity 3 to 5 end acts as template strand. The other strand which has polarity 5 to 3 end and the sequence same as RNA except thymine at the place of uracil is displaced during transcription. The strands of DNA which does not code for anything is called coding strand. Here in the given one example of template and coding strand is given. You may find here that template strands start from 3 to 5 end and coding strands from 5 to 3 end. And also if template strands have adenine, then in coding strands it must be thymine. Likewise, if guanine is present, then the template is present in the template strands, then cytosine will be in the coding strand and vice versa. As adenine is always pairing with thymine and guanine is always paired with cytosine. This is the figure showing transcription unit having promoter, structural gene and terminator, and also showing the template and coding strand with their polarity. In transcription unit, the promoter and terminator are placed at side of the structural gene, means the position of structural gene is in between. It is flanked in between promoter and terminator. The location of promoter is towards 5 end, that is upstream of a structural gene. promoter is a DNA sequence that provides binding sites for RNA polymerase and also defines the template and coding strands. If position of promoter is changed with terminator, then coding and template strand will also be reversed. The terminator is located towards 3' end, that is downstream, and it ends the process of transcription. So promoter starts the process of transcription and terminator ends the process of transcription. Now the next topic is about transcription unit and the gene. As it is clear to all of you that a gene is defined as the functional unit of inheritance of character from parent to offspring. The DNA sequence coding for tRNA or rRNA molecule also defines a gene. In case of eukaryote, a segment of DNA is monocystonic, but in case of prokaryotic cell, it is polycystonic. Now I want to clear you about monocystonic and polycystonic. Monocystonic means it contains the genetic information to translate only a single protein. But in case of polycystonic, it carries the information of several genes and are translated into several proteins. In case of eukaryotes, the genes are split. Later on, we will see that how the genes are split. Now, let us see about the types of RNA and transcription process. First, we will see about types of RNA. There are 3 different types of RNA. What are they? They are messenger RNA, mRNA, transfer RNA, tRNA, and ribosomal RNA, that is rRNA. All these 3 RNAs are required for translation process, that is for synthesis of protein. So let us see what are the functions of 3 RNAs. The messenger RNA provides the template. tRNA brings amino acids and read the genetic information, and rRNA play structural and catalytic role during translation. Now next is about transcription process in prokaryote. In case of prokaryote, that is in bacteria, there is only one DNA-dependent RNA polymerase enzyme that catalyzes transcription of all the types of RNA. RNA polymerase binds to the promoter to initiate transcription. That process is called as initiation. Then the RNA polymerase catalyzes the process of elongation. Why? Because only one enzyme is present. When RNA polymerase reaches the terminator region, there is termination of transcription, means transcription process stops. The process of initiation and termination are due to one factor. That factor is called as sigma and rho factor. Sigma also called as initiator factor and terminator factor. Means sigma is the initiator factor and rho is the terminator factor respectively. This given figure reveals about the process of transcription in bacteria. You may observe here the 3 processes, that is initiation, second one is elongation, and then the last and third one is termination. You may here see that sigma factor along with RNA polymerase begins the transcription process, and rho factor helps in termination of transcription, thereby releasing messenger RNA. Let us see about transcription process in eukaryotes. It is little bit complicated. as here 3 RNA polymerases are present and each have a specific role. The RNA polymerase 1 transcribe ribosomal RNAs, that is 28S, 18S and 528S. Whereas RNA polymerase 3 transcribes tRNA, 5S RNA and snRNA. snRNA is the small nuclear RNA, and RNA polymerase 2 transcribes the heterogeneous nucleus RNA. With the help of different 3 types of RNA polymerase enzyme, thus primary transcripts are formed, which contains both exon and intron, which are non-functional. As previously we have discussed about the split genes. So here the primary transcripts contains both exon and intron, in which intron is non-functional. So let us know about exon and intron. Actually, exons are coding areas, whereas introns are non-coding areas of genes. So they must be removed. So splicing is done to remove intron and join exon in a definite order. Then what is done? Capping and tailing is done in heterogeneous nucleus RNA. In capping, methyl guanosine triphosphate is added to 5' end of hnRNA, that is heterogeneous nucleus RNA. And in tailing, adenylate residues are added at 3 end in a template. The given figure shows about transcription process in eukaryotes. You may observe here that primary transcripts contain both exon and intron. So splicing is done to remove intron, and also capping with methyl guanosine triphosphate at 5 end and tailing with adenylate residue at 3 end. As a result, functional messenger RNA is formed. So children, this is all about today's session. Thank you. For any kind of doubts, please write your questions in comment section.